Good morning, students.
Good afternoon, students. Once again, welcome back to Zoom class. Today we are going to discuss about endosperm and types of endosperm. First of all, let me introduce endosperm. What is endosperm? Endosperm is a nutritive tissue that is derived from a triple fusion, right? Endosperm, it is a nutritive tissue that is derived from a triple fusion, right? So it can be designated by 3N. It is a triploid in nature because during fertilization, you know very well, this is a ovary, then ovary encloses ovule. We have discussed about number of ovule, right, previously. And this ovule encloses embryo sac. I'm talking about embryo sac that lies at the center of ovule, in which you find at the center, this is called polar nucleus. And the polar nucleus is always 2N, diploid in condition, right? And these antipodal cells are present at the bottom of the embryo sac, you know very well, it's antipodal region. I'm talking about this microvillar region. The microvillar region consists of an egg cell at the center, and just near the egg cell you can see synergy cell. Why I have drawn the structure in order to understand clearly. When the pollen tube enters, now this is an entry of pollen tube, right? This is a pollen drive that enters into microvillar region. Here that penetrates nucellar region, and this pollen tube consists of two nucleus. So one is a generative cell and the other one is a vegetative cell. Among generative cell, you find there are two generative cells that we see, right? And the first cell that explains it is a pollen tube. When this pollen tube grows and penetrates the nucellar region, I'm talking about this is a male gamete, right? There are two male gametes all involved in fertilization. The one male gamete fuses with the egg cell. It is known as the egg cell. It is haploid in condition, and another nucleus of generative cell fuses with the secondary nucleus. It is also known as polar nucleus. So, by the fusion of male gamete with the secondary nucleus, I mean the polar region, which are having diploid condition of cells, this is a diploid condition of cells, the polar cell or secondary nucleus, which is fused with the, the fusion of one of the male gamete in the embryo sac to form endosperm by the process triple fusion. Now, triple fusion is nothing but it is the fusion of one of the male gamete with the secondary nucleus or polar nucleus. That is called a triple fusion. By the process of triple fusion, endosperm is developed. Just I'm going to point out the development and the origin of the endosperm. Actually, the central part of the embryo sac is known as secondary nucleus, in which one of the male gamete is fused with the secondary nucleus to form endosperm by the process triple fusion. Students, this is a definition of endosperm. Endosperm is nothing but it is a formation of a triple fusion that encloses one male gamete and a secondary nucleus, which is a diploid in condition. Both of them may fuse to form as endosperm. Now, what is endosperm? Endosperm is a nutritive tissue because that supplies the energy for the development of embryo. Actually, embryo is developed from the egg cell. If egg is fused with one of the male gamete to form as a zygote, right? Actually, zygote develops into embryo, then embryo develops into radicle and plumule of the plant, right? So this is simply something about development of the embryo and the formation of endosperm. All right, I can show you exactly where this endosperm is originated. Let me show the diagram. My dear students, I am going to illustrate this is a plant pot that we have discussed in previous class, right? You can say all the parts of flower, right? You can see very clearly. Yesterday we discussed, topic has been discussed yesterday, right? This is a flower where you can see the development of endosperm. As I learned in previous class, right? Endosperm is originated from the oval region. This is a ovular region. My dear students, please listen. Then you can understand very clearly where I'm going to focus. It is a structure of flower where you can see the green portion is called, you know very well, it's a calyx. The calyx is nothing but a group of sepals called the calyx. Right? It is a calyx. It's the outer covering of the flower. Then followed by corolla. Corolla consists of number of petals. And stamens are there, androecium, gynoecium is there, and lower part of the gynoecium. I mean, I'm focusing the central part of the gynoecium is called as the ovary. The lower part of the gynoecium is called the ovary. This ovary encloses ovule. Then ovule encloses, as I left at the very beginning, there are two types of nucleus. One is the egg nucleus and the other is the pollen nucleus. Egg is found just near the microphylla region. If you take any ovule in which you can find the upper part of the ovule is called as a microphylla region. Microphylla region. And the lower part of the ovule is called simply, or ovule is called the chelachal region. Right? So these in between microphylla region and the chelachal region, you find polar nucleus or the secondary nucleus or a central nucleus. This central nucleus, this is a region where the formation of endosperm you can say, right? So this is a polar nucleus in which one of the male gametes is fused with the secondary nucleus or polar nucleus to form endosperm. Actually, endosperm, it is a triploid in condition because it's a 3 stage. Because how it is formed by the fusion of polar nucleus is a 2 stage, and one of the male gametes, two male gametes are released from the pollen tube. One male gamete fuses with the egg cell to form as a zygote, and another male gamete or pollen tube fuses with the polar nucleus. So this is a polar nucleus and one of the male gamete which may fuse to form as a triple fusion. By the triple fusion, we can get endosperm, right? So this endosperm, what is the nature of endosperm? Endosperm is a nutritive tissue that develops from the central part of the ovule because it occupies more area in the picture that you see, right? Because it's a triploid in condition or 3 stage where you find this the central part of the ovary is occupied by simply, I can say, triple fusion, right? Endosperm is a further classified into following types, right? So this is a nature and the location and the origin of endosperm, right? So this endosperm which supplies the energy for the development of the embryo. Actually, embryo is surrounded by a specialized nutritive tissue that is called the endosperm, right? I can say, what is endosperm? Endosperm is a nutritive tissue that surrounds embryo which is formed inside. If you take any ovule, any seed in which you can find embryo is formed at the center, around which you can say simply endosperm. The endosperm is a nutritive tissue that surrounds embryo, right? So simply it is a nutritive tissue and it also regulates the structure on the development of embryo, right? During seed germination, right? So this is simply something of endosperm, right? So next we pass on to types of endosperm. My dear students, next we pass on to types of endosperm. Before that, I'd like to show you the structure of endosperm. You can clearly understand. In maize, you can find the structure of, right? I can say this is a structure where you find it is the structure in which you can see endosperm. So it is a seed code. This is an endosperm region. Endosperm. And at the center you find it is an embryo. Right? This is the embryo. This region is the embryo. This embryo is protected by specialized tissue, native tissue. It is called as a endosperm. If you take a maize plant, Siam maize, which you can find very clearly, the structure of endosperm. Actually, in the seed of maize, you can see the maximum area is occupied by native tissue. That is a endosperm, right? And one more diagram I'd like to show you. When you take a LS of the maize or Siam maize or corn plants in which you can find, this is a structure of endosperm, right? So the endosperm, which is seen very clearly as I labeled previously, right? This is a endosperm in case of maize, which is occupied by maximum area of the seed of maize, Siam maize that we can find, right? And the central part, this is the embryo. It is a embryo. So this embryonic axis is surrounded by a nutrient tissue that is called as a endosperm. So hoping that students, my dear students, you can understand very clearly the place where the endosperm which is occupied the structure of seed, right? The next we pass on to types of endosperm. My dear students, next we pass on to types of endosperm. Endosperms are classified into following types. The first type is cellular. The first type is nuclear endosperm, and the second category is cellular endosperm, and third type is kilobial endosperm. Then fourth category is ruminant endosperm. No endosperm, it is the following types, right? So the first type is nuclear endosperm. The first category is nuclear endosperm. The second type is cellular endosperm. And third type is kilobial endosperm. And fourth type is ruminant endosperm. Students, listen. Endosperms are broadly divided into following types. First type is a nuclear endosperm. Actually, nuclear endosperm is a type of endosperm in which there is no cell wall formation, right? All the nucleus, or simply you can say, this is a structure of nuclear endosperm. My dear students, it is a nuclear endosperm in which this is a nuclear endosperm. So simply this triple fusion or the endosperm or primary endosperm or PEN, primary endosperm. Primary endosperm nucleus. Actually, it undergoes several mitotic cell division. Hence, there is a 3N stage of primary endosperm nucleus mitotically undergoes, mitotically it divides into number of cells without wall formation. So there is no wall formation. Here you can see the nuclear endosperm simply the endosperm which undergoes several mitotic division. And there is no wall formation. There is no wall formation. Here wall is not formed. No wall formation. And all the nucleus are free. Right? This is the nature of nuclear endosperm. I repeat once again. Endosperm, it is of several types. First type that we discussed, nuclear endosperm. Simply a 3N condition of a primary endosperm nuclear cell division without wall formation. Hence, all the nucleus. This is a nucleus. This is a nuclear division. No nuclear divisions. Nuclear divisions only taking place, but without wall formation. Such a condition is said to be a nuclear endosperm. Right? I repeat it again. Nuclear endosperm is a type of endosperm in which it undergoes several mitotic cell division to form a number of nucleus, but without the formation of wall. So hence all the nucleus are free, including inside the endosperm. This is identified in case of coccinea. I can say some example for this category, coccinea, and one more category I can say, capsulis, capsula. The next category is arachis. One more example I can say, arachis hypogaea, arachis hypogaea. Arachis hypogaea is nothing but it is a binomial of groundnut. Groundnut, you can say this type of endosperm. This is a nuclear endosperm. Actually, nuclear endosperm is characterized in the case of arachis hypogaea. It's a binomial of groundnut. Coccinea, it is a binomial of ivy gourd, and a capsula. It's a kind of mustard variety. In these cases, you can find nuclear endosperm. Simply, I can say nuclear endosperm is a type of endosperm in which there is primary endosperm nucleus undergoes several mitotic cell division to form a number of cells, but without formation of wall. Hence, all the nucleus are freely floating inside. So this is called a nuclear endosperm, right? And next we move to next topic. That is a cellular endosperm. Actually, cellular endosperm is a kind of endosperm in which this is a primary endosperm nucleus undergoes several mitotic cell division to form a wall formation. You can see wall formation, right? Now let me illustrate. This is a structure of cellular endosperm in which you can find. This is a structure in which it is a structure of a cellular endosperm in which you find a number of cells with wall formation. So that is a major difference between cellular and nuclear. My dear students, this is a type of endosperm that is a cellular. As the name suggests, cellular endosperm. It's a type of endosperm in which, as I said, the primary endosperm nucleus, when it undergoes several mitotic cell division, you can see number of cells which are found inside of the primary endosperm nucleus. So hence you can see number of cells are closely associated. So such a manner is known as the cellular endosperm. The cellular endosperm is found in some cases. I can say some example. Adoxa member, and next one more member, that is a Helianthus. And one more category, that is a Scoparia plant. Helianthus, you know very well, it is a binomial of sunflower, right? In sunflower, you can see the seeds of sunflower in which you can find a cellular endosperm, right? So this is about the cellular endosperm. Next, we pass on to helobial endosperm. Actually, helobial endosperm is a type of endosperm in which there are two regions that you find very clearly. The one is a microfilar region and another is a cellular region. In microfilar region, the PN stage, I mean the primary nucleus, this is a structure in which you find it. It's a microfilar region. In the are free, each one is known as a nucleus. I mean, this is a endospermous nucleus. During mitotic cell division, I mean the primary endosperm, or shortly I can say PEN, a primary endosperm nucleus, when it undergoes mitotic cell division to form a number of cells. So most of the cells are present in the microfilar region. Actually, microfilar region, it is the upper part of the embryo sac. If you take embryo sac, there will be three regions you can see. The upper part is called as the microfilar region, where you can find a number of nucleus which are floating, which are freely originated without cell formation. So here in upper region, you can find simply nuclear divisions only without wall formation. Whereas in Chalachal region, this is a Chalachal region in which the primary endosperm nucleus undergoes mitotically to form a number of cells with cell wall formation. So here the major difference between a helobial endosperm, in which the microfilar region, it does not form any wall in between the nuclear division. Whereas in Chalachal region, where you can see a number of cells inside of the endosperm with wall formation. So that is the difference, right? Between microfilar region and the Chalachal region in case of helobial. So this type of endosperm that we can find in some cases, I like to mention helobial endosperm. So these members are mostly found in aquatic member. I mean, hydra plant. The hydra, and one more category I'd like to mention, Wallisneria. This is a two-member in which you can find the kilobile endosperm. The kilobile endosperm is a kind of endosperm in which you can find free nucleus and the malformation of nucleus. This is a free nucleus are floating in the region of microfilar region where you can find that all the nucleus are freely floating. They are moving from one place to another place. Whereas in lower part, the gelatin region, you can find it with the malformation of division that you can see, right? This type of nucleus, ulobile endosperm is identified in case of Hydra and the Wallisneria. The last type is the ruminant endosperm. Actually, ruminant endosperm is a characteristic of Carica cetaceae, right? So for that, I would like to mention, I would like to illustrate some diagrams to you. You can understand very easily. And one more example I would like to show you. Please understand. Students, it's very easy to understand the types of endosperm. And seeing the diagram, you can understand, right? When I take a cross section of the Aricaceae family, you can find this kind of endosperm, right? This is called ruminate endosperm. What is a ruminate endosperm? Actually, the endosperm cell, this is a structure of the endosperm cell. I'd like to show you. I'm talking about this is the endosperm cells in which the inner wall grows towards inner. The wall of the cell which grows towards inner side, hence it forms like, right? So the growth of the wall is irregular in nature. So hence this nutritive tissue which is supplies, which is spreads out throughout the cells of some cases, right? This is called, it's nothing but it's the ingrowth of the wall. It means there is a finger-like projection that arises from the inner wall of the cell, right? Hence it forms like a ruminate. So this ruminate, simply it is, I can say, marble in nature, right? Mosaic is also known as mosaic, right? And sometimes this kind of endosperm that I can say, Arica. Now simply the first category is Myrtaceae. Myristica. Myristica is one of the category, and one more category I can say. That is Arica. Catechu, right? Arica, Catechu. The next one, Passiflora. The next category is the Passiflora. So these are the members in which you can find ruminate endosperm. So far we have discussed about the types of endosperm, right? Students, it's very easy to understand. Endosperm, it is the following types. Please remember nuclear, cellular, kilobile, and ruminate. You can compare very easily on seeing the diagram. Nuclear, in which you can say there is no malformation. There is a cellular. You can see a number of malformation that you see in the structure. And kilobile, you can see two types of cells: the free nucleus and the associated nucleus. And ruminate, you can say the inner wall is only grown inside, right? I mean, the inner wall of these form like a projection, finger-like projection that grows towards the center of this thing. So this is a types of endosperm, right? And next we pass on to what are the functions of endosperm. Students, next we pass on to functions. What are the functions of endosperm? First, as I mentioned, it is a nutritive tissue that supplies the energy for the development of embryo. It provides, that means it gives a nutrition to the development of embryo. Actually, embryo, it is a part of the embryo sac that lies at the center. In order to grow, there is embryo which gets nourishment from the neighbor tissue. The neighbor tissue is nothing but now this is a embryo endosperm. This is the endosperm that supplies energy for the development of embryo. Right? This is a embryo. The embryo is originated from the 2N stem, zygote. This embryo which gets nourishment from the cell, right? This cell is called, this is a endosperm. Actually, the endosperm, which is a nuclear tissue, which is very large and it is occupied maximum area of the seed, in which embryo which gets nourishment from the endosperm. Second point, it also regulates the structure of embryo. It also regulates the structure of embryo, right? The shape of the embryo is determined by the presence of endosperm, right? In almost all the endospermic category that I can say, endosperm divides first. Endosperm undergoes cell division, and later on embryo develops later. So simply embryo develops, it starts develop division later, and endosperm develops first. It undergoes mitotic division to form a number of cells in order to grow and develop this embryo, which is found at the center of the cell. So this is the functions of endosperm. So next we pass on to embryo. Before that, I must classify. Seeds are two types. Based on the presence of endosperm, they are albuminous seeds and non-albuminous seeds. The seeds are divided into two types. What are they? The first type is albuminous seeds. Albuminous. I'm talking about classification of seeds based on the presence of endosperm, right? Albuminous seed, it is also known as endospermous seed. The second type, non-albuminous or X-albuminous. X means absence of protein. The absence of endosperm, X-albuminous seeds. And it is also known as non-endospermous seed. Non means absent, non-endospermous. What are the difference between albuminous seeds and non-albuminous seeds? Simply, this is a structure of seed in which you can find. You can see at the center, this is a endospermous seed, right? The presence of endosperm after development of seed, after development of embryo. Actually, embryo which gets nourishment or takes all the food from the endosperm. So after development of embryo in the seed, the endosperm, it is going to be retrained. Such a seed is a albuminous seed. Or simply I can say the seed which possesses endosperm is a nuclear tissue, even though the development of embryo inside of the seed. Such a seed is said to be as an albuminous seed. For that I can say some example. Paddy. And one more category they can say castor. So these are all the example of albuminous seed. I repeat once again. Simply presence of endosperm in a seed is said to be as endosperm seed because the term endosperm refers presence of endosperm. What is endosperm? Endosperm is a nutritive tissue. It is retained even the development of simply embryo, right? It is also known as an albuminous seed, right? For that, I can say some example: paddy, castor, right? They are the example of albuminous seed. Students, the next time it is very easy to understand. X albuminous. The term indicates X. X means the absence of. X indicates simply absence of albuminous protein. I mean, this endosperm is completely absorbed by the development of embryo. If you take a seed, now this is a structure of seed. I like to draw the structure of seed. Right? In the structure, you find this green color indicates presence of endosperm during germination. The nutritive tissue, it is completely absorbed, hence it does not possess. It is an endosperm, it is an embryo. Right? When this embryo starts developing in a seed, simply it gets all the food material or simply completely. This is a protein is substantially absorbed by the development of embryo. Such a seed is said to be as X albuminous. Simply, I can say absence of endosperm, or it is also known as a non-endosperm. Non refers to absence of nutritive tissue. There is no amount of nutrition inside of the seed. In such a seed is said to be as non-endosperm seed, or it is also known as a X albuminous seed. Right? So this is a types of seed. Students, I repeat once again, seeds are classified into two types. They are albuminous seeds and X albuminous seed. Albuminous seed means a seed possesses, that means endosperm, whereas X albuminous seed means a seed does not possess endosperm. It is completely absorbed by the development of embryo during certification, during germination process. For that, I can say some example for that. I like to give some example for that. The first type is pea plant. In pea plant, and one more category I can say pea plant, and one more that I can say groundnut, groundnut. So they are the example of non-endosperm seed or X albuminous seed, right, students? It is very easy to understand very easily, right? Please make sure to understand very easily. Next we pass on to embryo developmental. There are different things that we have discussed, right? After fertilization, there are so many changes that taking place in your plant. That is first fertilization changes, and during fertilization, this egg cell fuses with the male gamete to form as a zygote. The zygote develops into embryo. I would like to show you once again to you the diagram. Once again, I'd like to show you where embryo is present. It is the structure where you find it. I'm talking about embryo. I'm going to show you embryo developmental stage. I'm going to show you embryo developmental stage. If you take a flower in which One of the male gamete fuses with the egg cell to form zygote. Then zygote develops to form a two cell. In two cell stage, zygote is a diploid in condition. It develops into two cell stage. Again, it is divided into, that means it undergoes cell division to form four cell stage. Again, it develops into 8 cell stage. Again, it undergoes mitotic cell division to form a globular protein. Again, it undergoes several changes to form heart shape. Then finally, it forms embryo. The embryo is developed from the process of fertilization, right? So, the egg cell, it is a haploid in condition. One of the male gamete fuses with the egg cell to form as a zygote. I repeat once again. The zygote is a diploid in condition which develops into, it undergoes mitotic division to form as a two cell stage. Again, it undergoes mitotic cell division to form four cell stage. Then finally, it forms a 8 cell to form gives rise to form globular structure. It's a globular structure. Again, it is converted to heart shape. Then finally, it is produced an embryo. Instead of several steps which are taking place in the development of the embryo, right? So it's time to discuss development of embryo in diet. My dear students, listen. The next we pass on to development of dihard embryo. Dihard embryo. Development of dihard embryo. Other than example, I can say Capsula pastoris. Capsula burst pastoris. It is a stage that you find it is a zygote. It is a zygote. Zygote is always a two-end stage. After fertilization, the egg cell is converted into zygote by the fusion of one of the male gamete. Simply, you know very well. What is zygote? Zygote is a cell that consists of diploid condition, diploid cell that consists of both male gamete and female gamete. One of the male gamete fuses with the egg cell to form as a zygote. Zygote is the first cell of development of embryo, right? So this zygote undergoes mitotic division. It undergoes mitotic division. Mitotic division to form a two cell. The one is a terminal cell. and another one is the basal cell. The terminal cell is the upper cell, upper side is found. Whereas in lower cell that you can see basal cell. So the zygote differentiates into two cells. The upper side cell is called the terminal cell, and the lower part of the cell is known as basal cell. Again, these two cell stage of zygote develops into, it undergoes again mitotic cell division to form another shape, another division to form a four cell stage. This is another stage where you find right. Again, it undergoes right to form short, I can say TC and BZ. Basal cell, TC means the terminal cell, or upper cell and lower cell. Again, it undergoes to form a four cell state, right? Cell number 1 and 2 and 3 and fourth cell, right? This is a basal vacuoles are created. Vacuoles are developed when this basal cell grows and develops, right? Again, this cell undergoes to form. The basal cell is stable, and the TC cell, the total cell, again it grows in a different shape. As I said, it's a basal cell. It is a stable in form. It is not divine. Instead, this is a four cell. This is a TC cell or terminal cell which undergoes to form number of stages, right? To form globular structure. Now the upper part is called embryonal mass. Embryonal mass. It is a mass of cell that gives rise to form cotyledon. The embryonal mass which gives rise to form cotyledon. The cotyledons are a leafy-like structure that is enclosed in a seed. So when you take any seed, you can see there are two types of seed. They are dicot seed and monocot seed. When we cut at the longitudinal direction of a seed, the seeds are split equally. That is called as a dicotyledon. The presence of cotyledon 2. So these two cotyledons are originated from the embryonal mass or the embryonal axis that arises cotyledons. Cotyledons are nothing but it's a leafy-like structure that arises from the embryonal mass that includes simply radicle and plumule, right? Okay. The next followed by, you can say, hypophysis. Actually, this embryonal mass, just below the embryonal mass, you can find hypophysis. Hypophysis. Actually, what is hypophysis? Hypophysis is a cell that arises just behind the embryonal mass that gives rise to form, you can say, a plumule at the top and radicle. Plumule, it is a region of a stem. The plumule is gradually developed into shoot system. The plumule of embryo gives rise to form shoot system. Stem portion. The green part of the plant is originated from the plumule, whereas the broad part or the geotrophic condition, that is the root, is also originated from the radicle. So both these are radical and plumule. I'm talking about radical. No, radical is a region where it is originated, where it is. So actually, this radical is originated from the hypophysis region that gives rise to for root system. So simply I can say a root of the plant is gradually developed from the radical. So both the radical and the plumule are originated from the hypophysis region. And this radical or this root system, it is also protected by root cap. Again, this is a root cap that you can see. So this is a root cap. So there are mainly three portions that originated from the hypophysis region. The lower part of the cell, basal, or it is also known as a suspensor cell. It is also called as a suspensor. Suspensor. Actually, the suspensor cells are responsible for development of embryo, and it also regulates the growth of the radical and the plumule, and it also pushes the plumule and radical portion, right? So this is the role of suspensor. I repeat once again, suspensor is a cell that produces nourishment, nutrition to the development of plumule and radical. In addition to that, it also helps in pushing out a plumule radical from the stage. The next condition that I'm going to show you, and the stage. This is I'm talking about third is the fourth stage. Next we pass on the fifth stage. The fifth stage, it is a heart-shaped in nature. Students, listen. It is a So radical, in between radical and plumule, you find the structure is called cotyledon. So these cotyledons are originated, as I said, hypophysis region, right? And you know very well, this is a suspensor region or a root tip or root calf. So the outer covering is called cycot. So the structure that you see very clearly, right? It is a root calf. Here the embryo or embryonal mass gives rise to plumule and radicle. Plumule is modified into shoot system and radicle is modified into root system. Now which you can find there are two cotyledons which are originated from the hypophysis region, right? So thank you for joining students. This type of structure that you can see, Capsella barstoris. Capsella bursa. Stories. It is nothing but it is one of the variety of mustard. It is a mustard variety in which all the developmental stage of naked embryo is taken. I repeat it again. Zygote develops into two cells. What is the upper cell and there is a basal cell. The basal cell is stable and upper cell develops into number of shapes. They are globular, arc shape. Then finally it forms an entire development of the embryo, right? So thank you for joining, students. So far we have discussed a number of topics, right? Anything else? So you can ask the students. It's time for interaction, right? Endosperm. First we discussed about endosperm, types of endosperm and structure of endosperm. And endosperms are following types: nuclear, cellular, globular, ruminate. And seed, it is up to albuminous seeds and exalbuminous seeds. And the embryo, what is embryo? The development stage of embryo that we see, right? And embryo development that we discussed. Anything else? Students, you may ask. Any doubt you can ask. My dear students, this is the topic that we have discussed. You can ask anything. Students, how many types of endosperm that we have discussed? Yes, students.
Sir, 4 types.
Very good. What are they? What are the 4 types?
Nuclear
endosperm,
cellular endosperm,
fallopian endosperm,
fallopian endosperm,
ruminant
and ruminate endosperm.
Very good. And the seeds, how many types are there based on the presence of endosperm? How many types of seeds are there?
2 types.
Two types of seed.
Okay. How do you differentiate albuminous seed from ex-albuminous seed? Could you tell us in few words?
Seeds with endosperm is called albuminous seeds. Seeds
Very
without
good. Very good. Keep it
endosperm
up. Very good.
X.
Right. Okay. In last structure that we see, a heart shape of the right endosperm is you see the structure in which how many parts are there in a development of embryo? In development of embryo, we see following parts. What are they?
Embryonal mass,
Very good.
hypophysis, suspension.
Hypophysis,
Suspensor.
suspensor cell. Very good, ma. Thank you. Thank you for joining. And all of you, please try to make an assignment and try to send it to your respective subject teacher. What is the question? What is endosperm and classify its types? Thank you. Thank you for joining. And try to make an assignment and send it to your respective subject teacher. Thank you.